An ANDA sponsor submitted a sameness package for a peptide generic. FDA came back with 14 questions. Not because the data was wrong, because the story was incomplete. Hello, I'm Raghuvar Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, where we help pharma and biotech companies to build analytically airtight sameness evaluations for complex generic molecules. For small molecule generics, sameness is relatively tractable. Same API, same dose, same route, demonstrate bioequivalence and move forward. The moment you are working with a peptide generic like octreotide acetate, that conversation changes fundamentally. FDA is no longer asking, is this the same compound? They are asking, can you prove it analytically at every structural level with evidence that survives scientific scrutiny? That shift in expectation catches many teams off guard, especially those making the transition from small molecule development into complex peptide API territory for the first time. The stakes are not abstract. A sameness package that cannot clearly answer those questions doesn't just earn a deficiency later, it stalls your program while the clock runs, resources drain, and competing generics gain the market position. What makes octreotide specifically demanding is its molecular architecture. This is a synthetic octapeptide, 8 amino acids, a critical disulfide bridge, and a cyclic ring conformation that is exclusivity sensitive to manufacturing conditions and storage environment. You cannot simply compare spectra and call it done. You are dealing with a molecule that can differ subtly in sequence, bond connectivity, and impurity composition. And each of those differences carries regulatory weight. Structurally similar is not the same as the same. That distinction, frequently underestimated by teams new to peptide generics, is exactly where programs stall and generate months of avoidable back and forth with reviewers. The analytical case for sameness starts with the primary structure. For octreotide, that means high resolution mass spectrometry, amino acid analysis, and tandem MS fragmentation, matching the B and Y ion series of your generic against the reference standard. Residue by residue, complete sequence coverage documented without gap is the baseline expectations. Any ambiguity in the fragmentation pattern raises the kind of reviewer questions that are entirely preventable with the right experimental design upfront. From primary structure, the analysis moves to disulfide bond connectivity. Octreotide's biological activity depends on a single disulfide bridge that defines the ring structure. Verifying that bond isn't a formality, it requires non-reducing conditions, peptide mapping, and ideally multiple orthogonal techniques to confirm both presence and correct positional connectivity. A less experienced team might report disulfide bond confirmed and move on. What FDA needs is clear methodology that rules out scrambled connectivity or partial reduction artifacts, supported by interpretable method independent data. Here is where lived experience matters. One program we supported showed an unusually complex impurity pattern suggesting partial disulfide reduction under specific storage conditions, something the reference listed drug did not exhibit. That wasn't a stability footnote, it was a sameness signal, identifying it before the formal stability study locked in the wrong trajectory changes the entire shape of the development program. We got it because we were looking for it, because we had seen that what impurity signature means in practice, which leads directly to impurity profiling, the analytical layer where the most meaningful sameness comparisons happen. This isn't about measuring what's in your generic, it's about building the side-by-side scientific argument against the reference. Known impurity variants need identification, not just quantification. Unexpected peaks require investigation, not just footnotes. The method used for your generic and your reference must be consistent, otherwise, the comparison itself is scientifically indefensible. What FDA reviewers want is seasoned, reasoned analysis. What you observed, why it supports sameness, and what you did when the data raised a question. Physicochemical and stability testing close out the characterization picture. Solubility, pH, behavior, optical rotation, and force degradation under ICH conditions each contribute something the others cannot, and none can be safely omitted. For octreotide, stability is a structural characterization tool, not a checkbox. How the molecule behaves under thermal and oxidative stress reveals degradation pathways and aggregation propensity. If your degradation profile diverges meaningfully from the reference, that is a sameness question, not a formulation one, and must be treated as such. A comparative force degradation study, designed and interpreted correctly, can prevent an FDA information request before it is ever written. When FDA reviewers often open a sameness package, three qualities determine how the review proceeds. First, orthogonal evidence, multiple independent techniques arriving at the same structural conclusion. Second, validated reproducible methods, not fit for purpose shortcuts but defensible methods with documented performance characteristics. And third, a clear scientific narrative, data that explains itself. A submission where reviewers must reconstruct your logic has already created friction. CTT ready documentation is not about formatting, it's about communicating scientific confidence through structure. The framework is demanding but coherent. Confirm primary structure without gaps. Verify disulfide bond connectivity with orthogonal methods. Build a rigorous comparative impurity profile. Support it with physicochemical characterization and stability data that speak to sameness, not just quality. Each layer reinforces the next. Miss one and the package weakens. Build them well and you walk into review with a case that holds. Complex peptide generics are unforgiving of analytical shortcuts. The team that clears sameness review without deficiency cycles or timeline loss are those who treated the submission as a scientific argument, not a documentation exercise. If you are building a sameness package for a peptide ANDA and would value a rigorous second perspective, we would welcome that conversation. And please reach out to our team and we would be happy to discuss your program further. Thank you so much for watching this session and see you in the next one. Until then, goodbye for now.